Skeletal muscles are attached to our skeleton and aid in voluntary movement through contractions and relaxation of skeletal muscle cells, which are often called muscle fibers because they're so long. Muscle fibers are grouped into bundles called fascicles, and there are many fascicles in each skeletal muscle. Skeletal muscle is under voluntary control. Hence, skeletal muscle does not contract unless told to do so by the nervous system. For a skeletal muscle to contract, the individual muscle fibers must be stimulated by a motor neuron, which sends an impulse away from the brain and spinal cord and elicits a contraction. This video will describe the events that take place at the intersection between the neuron and the muscle fiber, which is called the neuromuscular junction. This image depicts a magnified view of the neuromuscular junction. The end of the motor neuron, called the axon terminal, is in yellow. Located within the axon terminal are synaptic vesicles containing the neurotransmitter acetylcholine, or ACH for short. The sarcolemma is the term used to describe the plasma membrane of the muscle fiber. The portion of the sarcolemma directly across from the axon terminal contains acetylcholine receptors. These are proteins that bind or receive acetylcholine. Notice the axon terminal and the skeletal muscle fiber do not physically touch. They're separated by a small gap called the synaptic cleft. Now that we know the parts of the neuromuscular junction, let's look at how a signal is transmitted from the axon terminal across the synaptic cleft and into a muscle fiber. When the action potential reaches the axon terminal, it causes voltage-gated calcium channels to open in response to the change in membrane potential. Calcium rushes through these open channels, down its electrochemical gradient, and into the terminal. The increased calcium concentration causes synaptic vesicles to release acetylcholine into the synaptic cleft via exocytosis. Acetylcholine then rapidly diffuses across the synaptic cleft. Once at the sarcolemma, acetylcholine binds to acetylcholine receptors. These receptors are chemically gated ion channels. These channels are like locked gates, and they only open with the binding of acetylcholine. Once open, these ion channels allow sodium to diffuse into the muscle fiber and potassium to diffuse out of the muscle fiber. However, the influx of sodium is greater than the efflux of potassium, so overall, net positive charge flows into the fiber. This causes the muscle fiber's membrane potential to become more positive, or more depolarized. Initially, depolarization takes place at the portion of the sarcolemma directly across from the axon terminal. This local electrical event is called the end plate potential. The end plate potential then initiates an action potential in the nearby regions of the sarcolemma. This action potential is a wave of depolarization that spreads or propagates throughout the rest of the muscle fiber's sarcolemma. It is this action potential that signals the muscle fiber to contract. But recall that the synaptic cleft still contains acetylcholine, which is still activating acetylcholine receptors. To return the neuromuscular junction to its resting state, this acetylcholine must be removed. This is largely accomplished by the enzyme acetylcholinesterase, which is always present in the synaptic cleft. Acetylcholinesterase breaks down acetylcholine, and once the acetylcholine is gone from the cleft, the acetylcholine receptors close, and the end plate potential stops. To summarize, the neuromuscular junction is where electrical signals in a motor neuron are passed to a muscle fiber through the release of acetylcholine. Let's review the detailed steps involved in that process. An action potential arrives at the axon terminal and voltage-gated calcium channels open. The influx of calcium causes synaptic vesicles to release acetylcholine into the synaptic cleft via exocytosis. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors in the sarcolemma. Acetylcholine binding opens these chemically gated ion channels. Sodium and potassium both flow through these channels, causing a net influx of positive charge. This depolarization of the sarcolemma, the end plate potential, initiates a muscle action potential. The muscle action potential then propagates throughout the sarcolemma, leading to a muscle fiber contraction. Acetylcholine within the cleft is then broken down by acetylcholinesterase. Once the acetylcholine is gone, the acetylcholine receptors close, and the end plate potential ends. This returns the neuromuscular junction to its resting state.